Good afternoon to one and all present, and we welcome you back to the second session, or rather afternoon session of FDP Day 1. And today's speaker for the session is Dr.S. Jagannath. He is Assistant Research Officer in Pasteur Institute of India, Coonoor, Tamil Nadu. Sir holds a PhD in microbiology from Bharatiyar University. He also holds a postgraduate diploma in bioinformatics and an MBA in human resource management from Periyar University. He has 2 patents for his credit, and he has published around 27 articles in various reputed journals like Epidemiology Research, Development and Evolution of Diagnostic Tools, Plant-Based and Bacteriological Research. He has also authored 4 chapters in books and is also an editor for a few journals. He has deposited his findings in gene sequence deposits at GenBank NCBI. He also holds quite a few honors and is also a member of a few forums like Life Member of Tamil Nadu Science Forum, Life Member of Indian Association of Vaccinology and Immunobiologics. He is also audit and regulatory officer for various bureaus. So we would like to welcome him to today's session. Sir, we welcome you on behalf of Department of Biotechnology and the Oxford College of Engineering. Now we would like you to start the session, sir.
Okay. Good afternoon, my dear friends. And also, first I congratulate the convener to start this kind of program through that online. Also, I thanks to the team member, those who gave this opportunity. As I'm going to speak about vaccine development process, vaccine development process, and what do you call and productions. Are you able to see my screen, sir?
Screen share.
Sir, it is sharing. You just minimize this and share your PPT, sir.
Yes, I now just only started for sharing. Presenter.
Yes, sir.
Okay. Now you got now.
No, sir. You just minimize this tab, sir. Your Microsoft Teams. So minimize this and click the PPT. Whatever you are open now is this. How do you want to join your Teams meeting? That
now
tab.
Now I minimize, ma'am. Now
it is not at minimized, sir. Oh. Yeah, it is getting minimized.
Okay. Have you seen anything, ma'am?
No, sir.
No.
Can you stop sharing and one more time share your PPT, sir?
One minute. Then I'm going for stop. Okay, now we'll stop and then again present, ma'am.
Yes, sir. Share again, sir. And
okay, I open the share tray. I click that. Then I minimize. Now it's. You can see,
no, sir. It's not at you started sharing. You are still visible. Your video is still visible.
Okay, something now is coming, no?
Yeah.
Presenting.
Yeah, sir. Go for slideshow, sir.
Okay.
Sir, slideshow is not sir. Below that, next to that 82%, that button is there. No, sir. You click that. Like down, sir.
Down. Okay.
Down next to that.
82%. Yes, sir.
82. And before that, you click that.
Yeah.
Still.
like a glass bottle. Yes.
Sir. It's loading,
Now,
sir. I think.
now it's coming. Now it's coming, ma'am.
Yes, sir. Fine.
Okay, good. Once again, good afternoon to all. I'm happy to share about, happy to start my presentation. So my presentations deals about vaccine development, process, and production. As you can see that different kind of pictures here in that first slide. You can see the person is going for searching for something. So again, you have the small virus type molecule, and next the Pasteur Institute. So here Pasteur Institute, it's one of the leading institute for the vaccine production, especially the DPT group of vaccine and the rabies vaccine. So these are Pasteur Institute, what do you call it? It's an heritage structure. And also in the top, you should have seen that from some of the different phases involved for the vaccine development. One is exploratory and preclinical, phase 1 clinical, phase 2 clinical, phase 3 clinical, then approval, and finally is a manufacturing and post-marketing. Okay, so here vaccine. So vaccine, a biological preparation, it can administered through various routes such as injection or inhalation or oral, which stimulates immunity against an infectious agent. Whether it's intramuscular muscular injection or a mucosal immunization or oral immunization, it can stimulate. And also usually it contains a harmless variant of a pathogen. Either it is in a killed or is attenuated microorganism, or it may be a laboratory generator, so altered form of its toxin or one of its structure marker. So what is meant by toxin? So toxin, it is nothing but as it will able to cause a pathogenicity. So that toxin is going for altered by a method called as killed or attenuated or heat inactivated. Here immunity, so ability of living being, whether it's a human or animal or any organism, to resist and protect itself from the harmful effect of an offending microorganism or a toxin. So that is called as a immunity. So immunity means the ability of living being to resist and protect itself from the harmful effects of an offending microorganism or a particular toxin. The efficiency of the immune system, it lies in its ability is going to recognize the foreign invaders without causing harm to host own cells as well as tissues. Vaccines, it acts by allowing the immune system to recognize and memorize the harmful viruses or bacteria of their toxin. That is called as a memory response or memory response of immune system. It going to recognize and recognize first is going for recognize as a primary antigen antibody reaction, and memory mean is going to keep something as a memory cells. That memory cells, if again the same kind of toxin coming or enter to the body, invading to the system, is going for again neutralization. So that is called as vaccines here. And also how we are getting the much more immune response by the booster doses. Booster doses of vaccine boost the memory of immune system, especially the IgG, immunoglobulin G, and increase its ability to fight against the invading pathogen. So invading pathogens is nothing but as a, what do you call it? It is a any microbes, other disease causing microbes or pathogenic microbes. So once a person going for vaccinated, they got only in 1 international unit of antibody on that 0 day or 0th day. Then again going for the same person going with same vaccine as a booster dose, immediately that 1 international unit immediately going for rise to that 3. So that is also boost up. So automatically the IgG response is much more. So there's a booster doses of this thing. And coming to this slide, so going for what are the facts involved about vaccine? The major contributions that vaccine have made in saving countless, so infinite lives around the world. Every year, up to 4.5 million children's lives are saved by immunization as per World Health Organization. And also the vaccination is a proven and one of the most cost-effective child survival interventions. So this one type of intervention is one of the cost-effective especially for the child survival. And all countries in the world have that own immunization program to deliver selected vaccine to the targeted beneficiaries. Here, each and every country, the World Health Organization, they should not going to follow the each and every country is having that own immune program. So that country having that own government policy, and also where the country was located, what kind of disease going for provoking, what kind of populations they have, what kind of mortality and affecting rate, death rate. Based on that, that country is going to fix it as a own immunization program. In case India, we have the own program that is called as Universal Immunization Program. Previously, that is called as Expanded Program for Immunization. So here, the UIP is targeted to the particular beneficiaries, especially focusing on the pregnant woman. So pregnant mothers also going for vaccinated in that tetanus toxoid TD during the pregnancy. After the newborn baby is coming, that also going for covered with BCG, mumps, measles, rubella, different kind of vaccine you can see later. So who are at a high risk of disease preventable by the vaccines. Yes, vaccine preventable disease. It also comes under the Universal Immunization Program. That is our India's own program. Previously, it is called as a EPI, Expanded Program for Immunization. There are around 27 causative agents against which vaccines were available, and many more agents are targeted for development of vaccine. 27 causative agents are identified and the vaccines are identified and also approved in the entire world. But vaccine are not only preventing the infectious disease. Some of the vaccine it help the body to fight range of illness by activating their immune system. Active immune system to regain it and attack the diseases. To activate the immune system, we have the own immune system. That immune system would be in sometimes it would be in hiding, sometimes it would be in hibernation. So that type of immune system, once going for vaccinated, is going for elicit immune response. So that is active immune system to recognize and attack the disease. Okay, here I'll give some of the disease, vaccine preventable disease and the causative agents and also the vaccine. Here, Mycobacterium tuberculosis is a causative agent of TB and controlled by the BCG. Why BCG is more important? Why is giving to that if the baby was born immediately? Because so normally 99% kids are born in a hospital. Sometimes the hospital it is not focusing on the single type of disease preventing. So that hospital is having the different kind of disease patients come to the hospital if they are going to sputum off. The sputum containing that kind of bacteria, there is a Mycobacterium tuberculosis bacilli. That bacilli, it can be easily spreaded through the aerosol. Once the newborn baby is coming, I'm going for inhaling that kind of pathogen or invading system. So automatically it's going to entire the lung system going to affect to avoid that. So immediately the newborn baby born, so going for the vaccine with BCG as a single shot. That's a lifelong immunity. And next is Corynebacterium diphtheria, pertussis, tetanus. So these are the another causative agents to causing diphtheria, whooping cough, and also tetanus as well as neonatal tetanus to the newborn babe. So that all controlled by the DTP group of vaccine. DTP means either diphtheria, tetanus, or diphtheria, tetanus, pertussis. So this kind of vaccines are a booster dose also we are giving to the particular children. Next is a poliomyelitis viruses. It's causing polio disease, and also we can control by OPV, by oral polio drops, and also fractional inactivated polio vaccine. I will share one thing. So in India, around the world, only India, that polio countries has fully eradicated. So last poliomyelitis case was happened to India in January 13, 2011. From January 13 to 2011, still we are not received any kind of poliomyelitis disease. So that's why in 2014, the World Health Organization, what they done, they are declared to give a letter to India. So your country as a fulfill country, so eradicate the entire poliomyelitis. How it was happened? Yes, we people are one of the member to among the that can to achieve the goal. Especially for that, every year in the month of January and February, those kids at the age of below the 5, they are going for vaccinated. that vaccine not only from the hospital, it will be in bus stand or railway station, wherever the peoples are gathering. So some of the school children, college children, being as a voluntary, they are going for pulse polio drops. Because of that, in India, we are achieved as a eradication of poliomyelitis. But still, in 2 countries in the world, it's going for the polio cases. One is Pakistan, and another one is Afghanistan. If that country is going to eradicate the disease, that polio will be in one among the second disease under eradication list. So which one is the number 1 disease was fully eradicated? Yes, smallpox. So next is measles, and also measles virus is causing the disease. Measles we can control by the measles mumps rubella, then Hib vaccine. So Haemophilus influenza virus is causing the influenza disease. We can control by the Hib vaccine. Next, rabies virus controlled by the rabies vaccine, and also Japanese encephalitis viruses, and also it can caused by the Japanese encephalitis viruses. It is some of the country in our state, in India, some of the state having this kind of Japanese encephalitis viruses is sometimes it is the particular having that outbreaks. We can control with JE vaccine and also rotaviruses. And rotavirus is one among the booming and also is a highly mortality rate disease in the world. That's why that UAP it's taken the disease for rota vaccine under the UAP program. Here, so already told one person he was developed the vaccine. That vaccine called as smallpox. That only disease fully eradicated till date around the world. That is nothing but called as the great man, Dr. Edward Jenner. In 1796, he was tested his empirical knowledge and wild cattle disease cowpox to protect against the deadly protect against the deadly it is in that diseases here. And the next is the great man Louis Pasteur. In 1885, he was the development of rabies vaccine with spinal cord of rabies infected animal. You can see there's some photograph here. Nice. He was supporting something. He was holding the bottle. That bottle is having that rabies viruses. Inside that bottle having that rabies infected animal spinal cord. That spinal cord only he was taken and then dried in a sunlight. That sunlight dried vaccine he gave to that particular person is died. And secondly, that spinal cord is dried with sodium hydroxide. Sodium hydroxide, what we are using is a detergent. That sodium hydroxide dried spinal cord, he developed a vaccine. It was given to the person called as a Berger-Jubilee. So these are the great vaccinologists towards the development of different kind of vaccines here. Okay, that is some of the vaccine. Here, what are the vaccine ingredients? Vaccine, it is not a single component. It having the different kind of multiple components. Yes, vaccine contains a tiny fragments of the disease causing organism or the blueprints for making the tiny fragments. Either is a protein or is a DNA, is RNA or mRNA. So different, that is called as a blueprint of making the tiny fragments. And also it also contain other ingredients to keep the vaccine safety as well as effective. That is the ingredients. And also each and every vaccine, that component serves as a specific purpose and each ingredient is tested during the manufacturing process. All the ingredients are tested for the safety aspects. Antigen. So antigen is any kind of a toxide material. So the toxide is a protein or it is in some of the capsular protein or is in a, it is in a crown-like structure protein or is a G protein, L protein, different kind of protein or virus protein or bacterial protein. It can elicit immune response. There is an antigen. Stabilizer. So here stabilizer means that vaccines is going for a immunization purpose. Immunization during the immunization, before the immunization, and also we have the different kind of processing. That stabilizer, it can keep the antigen material in as such condition if you are going for -70° temperature or +37° temperature. During that temperature, that specificity of the protein, that means antigen, it should not lose their original activity. So that was protected by the stabilizer. And the next is surfactants. So the surfactants, they keep the vial is going for vaccine filled in the vial. So that vaccine, it should not be contact with that glass vials, otherwise it being sticky in nature. So we can use it as surfactant. And next is a preservative. So preservative means once a vaccine going for opened in a is, for example, it's a multi-dose vaccine. Then going for open and kept in a fridge plus 4 degree. In the fridge, there is a chances to that making of different kind of microbes in and around. So automatically it's going for contaminated. That means less than plus 4 degree growing bacteria. So that kind of things we can avoid that different kind of preservative are used. That preservative should tested without causing uh, any kind of problem to the person. Next is adjuvants. Yes, adjuvant sometimes aluminum hydroxide gel or calcium phosphate gel, or is an ISCOM gel, or it is a nano emulsion gel. So different kind of gels used as a adjuvant. So adjuvant, it is not a immunization factor, it is a cofactor. Cofactor to the antigen, it elicits higher immune response. Sometimes it's a human albumin as a adjuvant, and also aluminum hydroxide as a adjuvant. So these are the things like antigen, stabilizer, surfactants, preservative, and adjuvant. These are the main components for the ingredients of the vaccine. And next is a diluent. That diluent, it is more useful for the, especially for the freeze dried vaccine. The freeze dried vaccine in generally it is a viral based vaccine. Why the vaccines are freeze dried? The freeze dried vaccine is a prolonged, what you call prolonged storage capacity. For example, some of the vaccine it should be stored in a plus 4 degree. So the plus 4 degree is available, and some of the vaccine need a minus 20 degree. So that kind of things we can use for the this kind of diluent for keep the material in a frozen condition as a powder form condition. So once just we go into the hospital, what done? The doctor taking a diluent is a cut the diluent. The diluent always containing WFI. That means water for injection, and also it's a endotoxin free. It's a free from microbes. It should already passed with the different kind of GMP activities, and also sometimes a 54 millimolar sodium chloride. It's a normal saline. That normal saline is taken from that ampoule. Then going to inject to the vial, vaccine vial, going fully diluted. So diluted only, taking the injection, they're going for vaccination. Okay, then what is vaccine? What are the different types of vaccine? One vaccine is called, first is a live attenuated vaccine. This live attenuated vaccine means it is altered or weakened form of the microorganism which lacks virulence but retains the ability to create immunity against the disease. That means that pathogenicity was attenuated. So it can having the capacity of the immunogenicity. For example, MMR vaccine against measles, mumps, rubella, then varicella vaccine against chickenpox, then oral polio vaccine against poliomyelitis. And the next is inactivated vaccines. That inactivated vaccine is a live organism or killed and inactivated and used as a vaccine. They are usually heat stable and do not need exact temperature of storage. For example, pertussis vaccine. It is a wholesale pertussis vaccine, or the acellular pertussis vaccine is a part of DPD group of vaccine, and another called as a rabies vaccine, and sometimes is a co-vaccine of corona vaccine. These are the inactivated type of vaccine. And the next is what do you call it? A toxide based vaccine. So toxin is secreted from the bacteria, either diphtheria toxin or tetanus toxin. The toxins we're going for inactive by chemical or heating method. So that toxide converted as a vaccine. That is a vaccine. Okay, I'll give a small example. In diphtheria, so Corynebacterium diphtheria is a causative agent. In our Pasteur Institute, we have the PW8, Pox William 8 strain, that is a highly immunogenic and non-pathogenic. The strain was going for grow in a FDM. What do you call it? Is in a PDM. It's a peptide digestive media. Is going for propagated. After the propagated, that entire fermentation, we collected the toxin. So during this 36 hours, it secreted something in the fermentation. That toxin we are going for inactivated with formaldehyde of 0.7% for that 7 days. That entire material was kept under 37 degree for 57 days. Entire material was kept in 37 degree for 57 days. Why that too much of temperature? Why that time of duration? Yes, the toxin, it is a double stranded. One is diphtheria toxin is alpha and beta stranded. So that two strand having the combined with the disulfide bridge. That disulfide bridge going for added inactivating agent is a formaldehyde is cut the disulfide bridge, automatically the alpha and beta chains is separated. Once separated, that is called as a toxide. Sometimes that separated toxide is going for irreversible, again going to form as a toxin. That's why you get for 57 days in a 37 days, 37-degree temperature. So that is called as diphtheria vaccine, is a toxide based vaccine. And next is a using with a different kind of modern technique. Either it's a subunit vaccine, or it's a recombinant vaccine, or it's a polysaccharide vaccine, and a conjugate vaccine. These type of vaccines are produced by employing more modern technologies through for purifying and isolating the active components. Active components means they say it can able to neutralizing antibody. It's a highly immunogenic and also with the non-toxic, that is called as active components. As a protein, as a single protein, as a combined protein, as a capsular protein, is a polysaccharide proteins, or some of the carbohydrates. So that is component, active components of the microorganism which can able to produce immunity but not a disease. Just keep in your mind that vaccine means it can able to produce a immunity, not a disease. For example of subunit vaccine and the recombinant hepatitis B vaccine, and the next is a HPV. HPV is a human papilloma viral vaccine. In 2030, the World Health Organization, they are having the policy that HPV vaccine also going to the mandatory to the new mandatory to the girls, those who are attained the age of puberty. Okay, now we got some clear about what is vaccine, what are the different kind of vaccines, who are the vaccinologists, some of the vaccine preventable diseases. Now I'm going to the vaccine development. Okay, so here you can get some good information about vaccine development. Either is empirical vaccine development or the modern vaccine development. Empirical means it is from 1702 until around the 1980s. So here you can have the different kind of vaccine. One is a variolation vaccine, that is a smallpox vaccine, and a live attenuated vaccine, and also killed organism vaccine, and a polysaccharide vaccine. Next is a cell culture based vaccine. Yes, cell culture based vaccine, roto vaccine, rabies vaccine, polio vaccine, JE vaccine. So these are the vaccine called as empirical based vaccine development. Yes, the technique is a killed or attenuated or live or heat inactivated or purified form of this called as this kind of empirical vaccine development. And from 1980s onwards until date, we have the different kind of vaccine technique that is called as modern vaccine development. In the modern vaccine development, one is a glyco-conjugate vaccine. Glyco means as a what do you call it? So glyco is a carbohydrate and also conjugate based vaccine. Why the conjugate based vaccine? That is called the hemo plus influenza. Here the influenza, that bacterial strain is called the influenza diseases. So it is not a single strain, it's a multi-strain. So each and every strain having the different kind of prone A, prone B. There is a protein involved for the neutralizing antibody. That kind of things is useful for that causing immunogenicity. So what they done, different kind of strain of the particular bacteria, influenza, hemophilis influenza. So take the strain and also that strain going for some glycon and glucon, glucose, then going for conjugated like a binding that also vaccine. There is a glycoconjugate vaccine is a hemo plus influenza. And the next is a recombinant DNA. So recombinant DNA vaccine is called hepatitis B and the human papilloma vaccine. These are the one of the approved vaccines. And this another one vaccine is called as a recombinant Ebola based vaccine. And the next is reverse vaccinology. Now that reverse vaccinology and the structural vaccinology, it is a vital role in last 1.5 years, from 2019 November onwards. Yes, we people are sometimes what happened, we are gathered, we are in isolated in some houses, and also we kept in some as a lock because of lockdown. Either during lockdown, why it was happened? By the pandemic of COVID-19. So that COVID-19 viruses, so once it was happened to the China, immediately they are using the technology called as structural vaccinology and the reverse vaccinology. So normal vaccine means identifying the antigen, then going for propagation and identify the toxin then toxide conversion, then going for vaccine. But here, reverse vaccinology means which gene is responsible for particular disease. That gene they identified, then going for what do you call it? It is a synthetic amino acids, then going for proteins, then going for it is a vaccine. That is a reverse vaccinology. And the structural vaccinology means it is a what do you call it? It is a helical based structure, or it is in what do you call it? It's a structure based, the motif based structure. That kind of vaccinology only used will get a vaccine around, will get a vaccine in the world around within a 18 months. 18 months as a good vaccine against COVID-19. And next is synthetic seeds vaccine and immunotherapy for especially in the cancer patients. And next is nanoparticles. For the nanoparticle adjuvant liposome, these are the things used for the drug delivery or vaccine delivery or nano vaccine delivery. Either it is in a nasal based, or it will be oral based, or to be in a transdermal based. So different type of things used is a, these are the modern vaccine development. Okay, now in this picture you can clear some details about vaccine development, how long it will take. So the syringe, it will take here the infectious agent was linked to the disease, and here that vaccine was licensed in the USA. For example, influenza, we find out the disease in 1938. So after lot of research and development, 2004, that influenza vaccine will be implemented. But smallpox, we doesn't know where it was started, when it was started. Averagely, 3 of every 10 people who got it died on that day. So that's why in 1949, the successful vaccine was processed as per the concept was developed by the Edward Jenner. Malaria, we identified in 1880, still there is no vaccine. But many effective anti-malarial drugs are available. That drug also used for COVID-19 recently for repurposing of drugs. In tuberculosis, we identified in 1992, still there is no proper vaccination. And also typhoid fever, identified in 1884. In 1918 only, we got a very good successful results. Here, very good means it can cause safety, efficacy, potency. It should not cause any kind of adverse reaction. Like in 1889, we identified the meningitis. That meningitis, so that vaccine was identified in 1981. And the next is a pertussis. 1906, we identified that pertussis disease. In 1948, we got the good vaccine against pertussis, like DPT group of vaccine. Dengue, we identified in 1907. Still, there is no proper vaccine. But polio is identified at 1908 and 1955. There is a vaccine called against the poliomyelitis. Next, 1947, the Zika virus came. So still there is no proper vaccines. In chickenpox, we identified in 1953. We got a vaccine in 1995. But measles, so we 1953 we identified, but we got combined with mumps and rubella. Then mononucleus is in 1960. Still there is no vaccine. And hepatitis B, we identified on late 1965 or 1981. We got hepatitis B and A vaccines. Like rotavirus vaccine, we identified in 2006. And what do you call it? It is a cervical cancer, HPV vaccine developed by 2006. Still, we are not having that vaccine against AIDS and malaria and other kind of. This is how long it will take for the vaccine development recently. Okay. So here during the development, what are the various steps towards the development of vaccine? So is a development phases in the laboratory. Either it is in colleges or university or institution, a different kind of things, that kind of to identify the component. That is a lead molecule we are developed in a laboratory. And next is the developed material we are going to test it in a PCT, tested in a lab condition by the IVT, in vitro testing. In vitro testing means that vaccine should clarify the laboratory based experimenters won't then immediately going for the next test is called as preclinical PCT in a suitable animals. Then going for that successfully going for clinical trials in a human being and also approval from the regulatory body, and finally continued surveillance after marketing. So these are the various steps you can get clear in a next stage. Okay, discovery. So what is meant by discovery? So discovery of the particular disease we can identify it in a lab. So that means it's a root cause. In the root cause we can identify. So for example, some fever is there. So where it was, why it was happen. Either it's a causative agents, either it's a bacteria or it is a viruses. It's a pathogenic bacteria or it's a viruses. It's a root cause. It is from waterborne or it is a airborne or that kind of things. So then going for affecting rate, then going for mortality rate, and also there is a current measures. Based on this criteria, we can develop a vaccine candidate. Next, going for the bulk process, that is a process development, and bulk manufacturing, product finishing. The process development means to produce an economically viable vaccine consistently in a manner that satisfies regulatory. Economically viable vaccine, that is a bulk manufacturing process. What is meant by economically? Economically means, for example, if you are going to produce a vaccine, vaccines around 1,000 rupees, it's okay. It's going for 1 crore rupees cost of vaccine means, so that is a economically viable. So that is a affordable by the common man, and also consistently. So that means each and every vaccine development, we are going for consistent result in a 3 consecutive batches in a manner that satisfies regulators. So who are the regulators? As a national regulatory authority, as CDSCO or FDA or US FDA, and also we can going for that compendia of Indian Pharmacopoeia, US Pharmacopoeia, Japanese Pharmacopoeia, and European Union guidance. So these are the regulators. This is a process development. Next is a clinical development. Yes, I developed the process. I produce a vaccine. I tried in a PCT in a preclinical animal testing. Then immediately going for clinical testing in a human being. So clinical development means it is going for phase 1, phase 2, phase 3. In clinical development, to demonstrate the safety and measure the protective effect of the vaccine in humans. To demonstrate, it can be able to demonstrate. Yes, one single dose of vaccine going through vaccine to the person, that vaccine it can able to elicit an immune response. The same kind of immunogenicity it will be in another kind of person. That is a demonstration, and also it be in a safety. Safety means it should not cause any kind of adverse reaction like allergens, like seizures or death. So this kind of thing it should not happen during the clinical development. It is a phase 1, phase 2, phase 3. And next is a assay development. Why the assay was used? To check the immunogenicity, to check the vaccine potency, to check the vaccine stability, is to develop an appropriate test. Appropriate means, so the particular test, it can appropriate only the suitable of the vaccine to ascertain the purity. Purity means that vaccine should not contain any kind of other unwanted material. What happens if other materials be there? It will causing the adverse reaction, it causing the allergy, it causing seizures, it causing death. So to develop an appropriate test to ascertain the purity, so that's why we are development of ELISA kit, development of MAP kit, development of other kind of in vitro kit, other kind of in vivo kit. So assay development is potency and stability of the vaccine under development. So one is it can pause the purity and also pause the potency. Potency means the immunogenicity, and also challenge method. Next is stability. Why is stability is more important? The stability only we can fix the vaccine shelf life. Either it is in 15 days or it is 30 days or it be in 3 months or be 6 months or 2 years. So once a bulk was produced, bulk means it's a one small quantum of the vaccine that is going to the vaccine. That bulk I am going to keep in a different type of temperature by this ambient temperature is in 22-degree temperature, or it will be in stressed condition or 37-degree temperature, or like going to accelerate degradation by the -20. So I get the same vaccine, same bulk would be in various kind of temperature. So I'm going to take in the sample for every 7th day, 0, 7, 14, like that. So 7th day, I'm taking the vaccine from the various spaces, various storage temperature. I'm going to check the safety, check that integrity, check the potency, and I'll check the entire thing. if any kind of this specific activity was the intended by the regulators, you should adhere with that. If which time on which condition that vaccine lose their safety or efficacy or potency. So that is their ultimate point. So once we find out the ultimate point, immediately we may going for that fix the vaccine should be in shelf life of only in 1 year or 2 years. So generally the liquid vaccine always the expiry period is only 1 year. As a freeze-dried component, expiry period is a 2 years. So this only you can get from the stability based record. On the next you can get the what are the phases, different phases in a detail about how many numbers of person we need, what kind of activity is there. So everything we can get in a various phases. Previously we what we find is a process based, as a phases based. Yes, in PCT establish a vaccine candidates as desired effects. You can give that. So what language I taken you, everything I taken from the World Health Organization is a regulatory terms. So establish a vaccine candidate as desired effects in vitro. So as a in vitro and laboratory conditions, I am going to clear that in vitro, then effects produced consistently. It should be in three, what is the trial batches 3, and also validation batches 3, and also process qualification batches 3. So each and every batches I'll get a same kind of results. For example, pH will be a 7 means that's a pH always in the 7.0 means plus or minus 0.1. It should be in replicated from entire sequences. For example, potency 1.5 means it can be all times, and also going for conductivity 18.2 bego means like that. So everything would be consistently as a PCT. One, I got a very good result in a PCT trial immediately I'm going for in a animals. And also desired immune response, it can protect animals from a disease, ensure relatively free of side effect. So what is my free of side effect? So once vaccinated, so based on the vaccine, it can going to elicit immune response. If it is not that, if going to affect to the any kind of lung-based system affected, I'm going for thymus-based affection, kidney-based affection. This kind of toxicity should not allow, should not be there during the animal test. And also, if you are going to use the animal testing, we can choose the animals like is a mice or guinea pig or local mice or social, but any kind of mice. That also we can pre-approval by the approval from the IAEC, Institutional Animal Ethical Committee. Once we can decide we successfully got a very good result in a animal clinical trial, we may going for the human volunteers. That also CT clinical trials, either phase 1, phase 2, phase 3. Phase 1 means less than 100 healthy volunteers, escalating doses introduced. That means single dose or 2 dose or 3 doses is a escalating doses introduced in a 100 healthy volunteers. Safety assessment using blood sample. Yes, we are going for tested in a healthy volunteers. So every day we are taking the blood sample, we assess the vital signs. Safety assessment that vaccine it will not causing any kind of inappropriate reactions. Next, moving to the phase 2. In the phase 2, 100 plus volunteers intended for particular age as well as diseases, and additional safety efficacy test immunogenicity studies to capture the immune response. Here only the immunogenicity capture. Okay, what kind of seroprevalence was happened, what kind of seroconversion it was happened. That entire thing we can confirm in a phase 2. In a phase 3, 100K plus volunteers expand the safety databases. Here only we get a safety database. Each and every person results about their health as well as immunogenicity and also adverse reaction, everything it can fix it in a databases and to establish the protective efficacy and the clinical consistency. Then here only going for the market release, phase 3, and moving to the phase 4 as a post-marketing surveillance, detect the adverse effect, assess the long-term efficiency. So these are the some information about how the various phases in a vaccine trial. So next, moving for why the vaccine development, it will take a much more time. For reason one is, yes, less than 1/10 of vaccine candidate to achieve the licensure. So some of the vaccine it can failure in a in vitro level. Some of the vaccine it will come to the animal trial. Some of the vaccine it can pass in a phase 1 or phase 2, phase 3, it's going for failure. So 1/10, less than 1/10 of vaccine candidates achieve the licensure. The high failure rate is due to the unpredictability of the biological, yes, it's a biological organisms needed to produce a vaccine, and to the uncertainty of how human system will process and react the antigen. Some of the vaccine, it can be protect in a mice or guinea pig, but it is, we cannot expect from the human being. That's uncertainty. That uncertainty leads to the error of how the human immune system will process and react to vaccine antigen. And some vaccine candidate may produce appropriate levels of immune response, but it is induce important adverse reaction. It is produced. That means it is going for a safety. It is a failure in the safety aspects, but it will pause the immunogenicity aspects. And other vaccine candidate may be safe, but it's ineffective at preventing diseases. So this is about the vaccine development. So this is about the vaccine. One is a less than 1/10 of vaccine candidates to achieve the licensure, and also high failure rate is due to the unpredictability of the biological organism needed to produce a vaccine, and to the uncertainty of how the immune system will process and react to the vaccine antigen. Some vaccine candidates may produce appropriate levels of immune response, but induce important adverse reactions, failure in the safety. And other type of vaccine candidate, it may be safe, but ineffective to prevent the diseases. Okay, that is called as a vaccine development. Going for the vaccine processing. What is the vaccine processing? Here there is a upstream processing. Upstream means, so taken the antigen. What is there? So I'm going to develop produce a one vaccine. What do you call? I'm going to produce a one good vaccine and good strain. So I'm going to select the seed. Seed means what? Is here either it's a bacterial or it's a viral things. So it's a bacterial seed or it's a viral seed. If I'm going to produce a bacterial vaccine, I need a bacterial strain. If I'm going to a viral strain, I'm going for viral seed. Next, street, and next is a fixed. What is meant by street? Street means if the particular virus is isolated from the street by the epidemiological studies. That is called the epidemiological study. Some of the area we can target, for example, we may go to the Shimoga. Shimoga is more famous for the Siveri, Tunga, Bhadra is there. But Shimoga is always famous for the KFD, Kyanosis forest disease. Yes, there is a particular monkey, some of the tick is going for transmit the particular disease there. So we are going to take in that viruses. So that viruses was isolated from that animal is a street viruses. That street viruses, we does not know their incubation period, infectivity periods, and also how it's going to react that. The same street viruses, again, I'm going for converted as a vaccine candidate by the process called as fixed viruses. Fixed means the same street viruses I'm going to passaging or subculturing in a animals or suitable cell line. Either it is a virus cell line, or is a BHK21 cell line, or is a DHP and MDBK cell line, or MDCK cell line, or liver cell line, different kind of cell line. That virus going for infected, so infected viral virus going for kept in a separate tube, then going for again infect to the new monolayer cell lines, again infected. That is a passaging or subculturing. Why this passaging we need? During the passaging, that automatically that pathogenicity, that is a causative point. That pathogenicity automatically get down. First time it's going to titrate 10 to the 10 is going for 10, then going for drastically reduced to the 10, 9, 8, 6, 5, then going for 0. So once the pathogenicity getting down, automatically that immune response, its ability to develop a neutralizing antibody, that thing is going for increased. In a particular passaging, is a less immunogenicity and higher antibody less pathogenicity, higher immunogenicity. That is called as a fixed virus strain. That fixed virus only we can going for vaccine production. When a street virus means it does not do the incubation period, infected titer, and also how this bigger. But fixed virus, we know, we come across by the what do you call it in subculturing. We know the incubation period, what kind of infected titer, how long it's going for the multiplication, everything fixed. That fixed is going for the fixed as a non-pathogenic and also highly immunogenic. So the seed selection going for that. Then immediately I'm going for seed revival. So revival of seed. So before that, that seed always stored in a suitable cryoprotectant, either being liquid nitrogen, minus 197 degree temperature, or vapor phase, or minus 80 degree temperature. That seed I'm going to revival in a suitable culture medium. Either it is for minimum essential media or in bar anchorage dependent media, and also it going for that additives based media. So going for suitable media, then going for propagation. Once we got a selectable media, then there's a culture parameters. Immediately I'm going to the inoculate of the cell line is going for multi propagation. So propagation of the viral. That virus is going for by the fermentation with the critical culture parameter. Why I put the criticality? Criticality means, so I'm going for 100 liter fermenter. So I put a suitable medium, culture medium, and along with that fixed virus strain, and it's a fully characterized strain. So I'm waiting for 48 hours. During the 48 hours, that cell line, that virus going to propagation in microculture of something. It's a virus propagation. It should not free from any other microbes. It should not have the contamination. It should not should be in pH 7.5. It should be in DOT level very less. This kind of critical culture parameters, everything was fixed that soil got propagation. These are the things called as upstream. So in vaccine process, upstream means is a selection of the seed, either it's a bacterial or it's a viral. It should be in street. It should be a fixed virus. It is a non-pathogenic and highly immunogenic. I'm going for a revival in a suitable culture medium. Then propagation is a fermentation and bioreactor. So going for a critical culture parameter. Then I'm going to the harvesting. So I harvest the culture parameter. Now that virus was in a 100 liter culture vessels. Then the harvesting without contamination. Next I'm going for the downstream. It's around 100 liter material is there. So 100 liter it having the only 1 liter. That is a vaccine. How can I going for remove that? By the process called as clarification. That means free from contamination. For clarification, what kind of material I'm going to use? Either 0.45 or 0.22 micrometer of membranes. Either it's a polyvinyl difluoride or polyether sulfone or cellulose sustain membrane. There's a clarification to filtrate if the chances chances some of the times it may be in sometimes 0.01%, sometimes maybe get that contamination. To avoid that, we may going for clarified by the 0.45 and 0.22 micrometer level. Then I am going for concentration. Concentration means that 100 liter material, then I am going for concentration by the system called as UF. That was a filtration clarification is ultrafiltration. Ultrafiltration based on the molecular weight. If I'm going for protease vaccine production, so I'm using only 0.45 ultrafiltration, 0.45 microfiltration system. If I'm going for tetanus, I'm going to use a 30 kilodalton of ultrafiltration system because that tetanus molecular weight is above 30 kilodalton. In case of diphtheria also, I'm going to use with 30 kilodalton. But in rabies vaccine, I'm going to use only 100 kilodalton. Because that 100 kilodalton is the maximum the viral protein molecular weight is above 100 kilodalton. By I'm using the concentration. Next TFF. TFF nothing but called TFF is a tangential flow filtration. For example, it's a cassette. I'm going for flowing something that cassettes having the molecular weight cutoff limit is a 100 kilodalton. Those who are having below 100 kilodalton is a coming down. Above only is a retained. So that is called as a concentration. Once the concentration 100 liter of upstream material, so clarified, then concentration to 1 liter, then 1 liter material I'm going for inactivation with a suitable either a chemical based inactivation or heat-based inactivation. If I am going for chemical tetanus means tetanus and diphtheria, I am going to use with a formaldehyde by 0.7%. In case of pertussis, that whole virus I'm going to heat with a 50 degree temperature. And in case of rabies, I'm going to use with a what do you call beta propiolactone as a chemical base. So inactivation, so that was ensured in animals, then going for purification. That purification, so why we need purification? Some of the host cellular protein or host cellular DNA. Where the host cellular protein DNA will be happen? Yes, I'm going to propagate in a viruses for COVID-19 viruses. So during propagation, I use one cell line, for example, vero cell line. So that vero cell line, it can able to having that whole cellular host cellular protein and host cellular DNA that also causing from tumorigenicity that I should remove by the by the zonal centrifugation or begin ionization chromatography or is it affinity chromatography. After the purification was over, then I'm going to formulation. And I already told, so human will be in maltose or any kind of surfactant, stabilizer, preservative, cryoprotectant. So that this kind of things I'm going to add it in a suitable level, appropriate level, intended by the regulator. Then formulation with the suitable stabilizer and additives. Then I'm going to fill and finish. Finally going for freeze drying. So these are the different process involved in a vaccine production. One is a upstream, another one is a downstream. And the next is once during the processing is upstream down processing, I should have adhered with that quality control department norms as well as quality assurance norm. Next, I'm going for the IPQC testing. What is meant by IPQC testing? In process quality control testing. What kind of IPQC test I'm going to do there? Safety, efficacy, potency. Here is one process. For example, cell to the final vaccine production. Each and every process, each and every stage of process, it should. Hello. Just you should. I will complete my things. Then question hours. You can ask a question, ma'am, please. Hello.
Sir, some disturbance in between. Some participant has unmuted.
Yes. Okay. Okay. IPQC testing. During. Okay. I'm going for taking the cell lines for the propagations in the cell lines to final vaccine. One is a propagation, cell line propagation, and virus propagation. Then harvesting. Then concentration, inactivation, purification. So each and every stage, I should ensure the quality attributes of the material. So that's why I'm taking the quality samples by the QC team. So that was assurance of the safety, efficacy, potency by the IPQC in process quality testing that was done by the production team. And also QC, I'm going to take in the sample. I send the sample to the QC. What the test they are going to do? Same what I have done in our production laboratory, like safety, efficacy, potency. So everything is completed, then going for random sampling. Yes, I have vaccinated my vaccine, completed the entire productions. So it being ready for the supply. So that QA people, quality assurance people, they come along with the QC people. They're taking the random sampling along with what results I obtained from the production, along with what results from the QC. They combine the results. They're taken that samples along with the results. They're sent to the regulatory. Who are the regulatory? Yes, Central Drug Laboratory. There's a direct guidelines from the World Health Organization. They're going for regulatory. So they're also having some standard because we are producing vaccine by following Indian Pharmacopoeia. As per the Indian Pharmacopoeia standard, our vaccine is already fulfilled and completed in our production lab as well as passed in a QC lab. The same kind of result should be passed in their sample, their laboratory. Once passed, only immediately going for the what do you call it? It's going for releasing. So these are the national regulatory testing for external validation. One is IPQC testing. During my vaccine production, I should ensure each and every stage of the production that material should meet the regulatory norm, should meet the regulatory, the safety, efficacy, potency, and also during the production process, I take some sample for external validation. I send to that material to QC. This QC is divided into different teams. You can see later. Then random sampling along with QC test result and the production results. They're sent to the serial. They are going for reanalyzed our vaccine safety, efficacy, potency for external validation. Finally, the vaccine going for releasing. Now you got clear. Okay, I'm moving for vaccine production. Just read up. One is what is vaccine, what kind of ingredients there, what are the different kind of vaccine, who are the vaccinologists, who are the vaccine, what are the vaccine protocol decisions, and what are the what types of vaccines. Then going for and what do you call process development, and also different type of processes and upstream downstream processes. Now I'm going for the vaccine production. Okay. So mandatory. So you can see what are the mandatory requirements for the vaccine production. You can see some of the pictures. One is quality assurance, one is quality control and animal testing, and the production is here. One is production, another one is processing. Next is the vaccine fields. So these are the things. How can we achieve that? By the mandatory requirement. So one is a facility. What is meant by facility? That facility means it should complies with the regulatory requirements. What are the compliance? Yeah, there's a building. So building, it should not have any kind of insect. It should not entering of the birds. It should not enter from the cattle. It should not enter from free from the unwanted material. And also water system. Water system means, so you know the different kind of water is available. One is potable water. One is raw potable water. Then that is going for pre-treated water. Pre-treated water means the water should have the multi MGF and ACF, the multi-grade filter treated water and activated carbon filter water and the softener water. That's a pre-treated water. That pre-treated water again going for the purification. Purify water by the reverse osmosis. Once the purified water was produced there, then I'm going for produce a water for injection. So the water for injection, where is coming from? From the pure water from the soft water from the MGF ACF, the MGF from that, MGF ACF water from that potable water. Potable water we may get from the municipal source or the well source or the stream source. The water should meet the regulatory norms. And next is a HVAC. HVAC means heat, ventilation, air conditioning system. If I'm going to a room, it's if in a final room, it's final filtration rooms. If final filtration room, it should be meet with 100. That means it should be in class 1. Class A is 100, then class B, C, D, E. So 100 covered with 1000, 1000 with 10,000, 10,000 with 1 lakh. If I'm going to a 100 class room, just I go to the 1 lakh, then going for 10,000, then 1000, then 1. So in a normal room, it comes under the 1 lakh. So 1 lakh, you can wear that coat and everything that. But another room, we can with the B government or C government, finally going the A government. So why it needs means to remove the particles. What is meant by particles? Either is a live, viable or non-viable particle. Okay, viable particle is a bacteria or viruses, any kind of thing. So viable, it is going to produce some cross contamination. What is non-viable particle? If I'm going to enter to the lab, I should not cover with my what do you call it, dust, that dongri dust and cover face. We should not with goggles and enter thing. Something or person in my hair, because I am not the headgear. So something the scalp or something hair is there. The hair is going to fell down in a floor. What happened? That floor with that floor. So it been attached there, immediately some of the viable material is going to the attached surfaces, it making the area as a contamination. So that's why I should follow the governing, I should follow the steps from that particular department. So the entire heat ventilation should be in, and also that what do you call it, is an airflow. Airflow should always in a particular positive airflow always there. Why we need a positive airflow means should avoid the accumulation of the viable or non-viable material. So flow pattern will be there. So once airflow is there, automatically suction will be there. So that flow I'm going to monitor in a BMS, building monitor system. Building monitor system means, so different kind of rooms there. One is corridor is there, normal corridor, dirty corridor, or change room, and my filtration room, and my preparation room. So different rooms there. So everything, I should not go and watch from each and every room. I should get entire detail from the top floor as a building management service. I get the entire information. What is the PA? What is that DP? And also what is that temperature? What is that? What is that? RH factor? Everything I can get information from the BMS. And next is a electrical. So electrical, we need uninterrupted power system. Why? So it's 24 hours my fermentation process. During the 24 hours process, I kept this as 600 RPM, pH 7, and also DOT is a 0.02, and also CO2 is a 0.5 percentage, and also conductivity is a 18.2 megohms. Everything would be supply as like alkali, acids, everything supplied. If current it shut down for 1 to 10 minutes, what happened? There is no vibration, there is no peristaltic movement, there is no heating, outer jacket. So everything happened. So we could not decide that. We put different type of chemicals and everything, same person, but it leads to contamination. Contamination means it is a sterile, it should not have any kind of material, but it is not passed because it is not release a particular toxin. So we need uninterrupted power supply. Then we need to the steam. Steam, either is a plant steam or is a pure steam. Plant steam, it is generated from that normal water, that is a pre-treated water. And pure steam bin, the pure steam was generated from that what you call pure water, that is the RO, EDI, reverse osmosis and electro deionization based water. That is a stream for jacket sterilization purpose, either in autoclave or decontamination autoclave or any kind of things used with this steam. Next is a effluent treatment plant and also sewage treatment plant, and finally we should have the bare house. Bare house means if I'm going to take a chemicals, I'm going to get something from what are the chemicals, raw materials we need, everything I'm taken in the stores, I kept in the production lab is a store condition. It leads to making of more viable particle because that entire chemical material stored in the cotton box, everything, that cotton box should not allow inside the production laboratory. So I should have the separate warehouse lab. I'm going to start a one batch process. I'll give the intendant. So peptone, this kind of things I need for 50 grams. I intend that everything they're pre-packed with the 3 packing system. So I've taken the system, I've taken the chemical, I wrapped other wrapped things. First wrap I'm going to remove that itself, and second wrap the class A, so class C, D, then class C, class B, like that. So these are the warehouse system. That's the facility. And the next, appropriate intendant instrument equipments. Appropriate. So in particular room, I'm going for DQ, design qualification. In that room, so what type of instrument I need? What kind of equipments? Is it a plug-in, plug-out instrument, or is it a fermentation, or a pH meter, or is it a conductivity meter, or is it a press, that is a food operated water system, or to be like that. So different kind of appropriate intended to the instruments and equipment for the particular room. Then we need a proper quality control department. So proper, these are the facility and the instrument is what's the facility. Then proper QCD. So proper QCD, different material department. One is the raw material testing. So each and every raw material should have the specific certificate of analysis and the material safety data sheets. Everything ensure that. Then going for in vitro laboratory. So in vitro we can using with estimation analysis everything in a in vitro, and in vivo using with animal experiment, and next to that microbiology block for the entire environment monitoring, and also to assess the microbiology aspects of water, microbiology aspects of steam, and the microbiology aspects of person, microbiology aspects of governing, microbiology aspects of eye. So everything will be tested in a microbiology department. Then going for stability. Once vaccine was fully completed, I checked the vaccine expiry period, everything. The stability is a vital role for the vaccine, especially for the releasing. Based on the stability record only, that vaccine going for releasing for certification. Next is a package material system. And next to the meet the regulatory norms. What are the regulatory? We need fire. So it should be have the fire around 60 particular water. It be in fully flowing to avoid the firing. You can see, you know that one thing. 1 month back, around a lot of course was damaged in Serum Institute because they are in, some of the fire failure in the in particular filling room. And next is a PCB. So PC means pollution control board. So pollution control board. So we are not we are producing vaccine. We are not having rights to the pollute that entire environment and entire water. So everything we are going to test it as per the regulatory guidelines from Central Pollution Control Board. And IAEC means Institutional Animal Ethical Committee for that proper animal using, proper breeding, and also what do you call going for euthanasia based. And finally, you need a qualified and a trained personnel. So these are the mandatory requirements for the vaccine production and along with proper quality assurance. So one is facilities, buildings, water system, HVAC, BMS, electrical system, steam, ETP, STP, warehouse, and appropriate intended instruments, equipments, proper QCD, like raw material testing, in vitro laboratory, in vivo laboratory, microbiology laboratory, stability, and package material lab, and to meet the regulatory norms like fire and safety, pollution control board, institutional animal ethical committee, then qualified and trained personnel for production as per the things, and proper quality assurance. These are some mandatory requirements toward the vaccine manufacturing. Okay, so you clear now. So I'm going to the how the vaccine was produced in our Pasteur Institute. What kind of mandatory things we are followed that. So before that, I'm going to explain about rabies vaccine production. Here, so rabies, what is rabies? It is a zoonotic disease that is transmitted from animals to human caused by the rabies virus. It's a Lassa virus within the family of Rhabdoviridae. And also this virus, it is transmitted through the saliva of the rabid animals and generally enter to the body via infiltration of virus-laden saliva from the rabid animal into a wound, like this scratcher or direct exposure of mucosal surfaces to saliva from an infected animal. And also is a 100% fatality. Is yet preventable. So mostly in dogs is a commonly as 95%, sometimes 4% in monkey, donkeys, horse, cows, buffaloes, goat, sheep, and pigs is a less than 4%, and occasionally 1% in wild animals, mongoose, fox, jackals, camels, and elephant. So rabies disease. And the next, what kind of things is rabies animal? You can see that some of the animals is a furious animal and paralytic rabies and rabbit raccoon. See that the dog is having some drooling of saliva, and another is a white color saliva is coming here. White color saliva is coming here near the gate, and there's a raccoon. This is a rabbit raccoon, and here dog drooling of saliva is coming. Is waiting for biting to others. And next is here is a rabbit, dumb rabbits. So these are some rabies animal. So rabies animals otherwise called as rabbit. Rabies infected person is called as rabbit person. How rabies virus was transmitted, especially the dog bite. So here the gentle dog, it will waiting for to bite the others. It containing white color saliva, and also I'll tell one thing. Based on this saliva color, we can differentiate. We can identify whether the dog having rabies virus or not. If the dog is drooling a saliva is a white color, means that is containing only few amount of rabies virus particle. The same saliva it being what do you call? It's an yellow color, pale yellow color. It leads the 50% of saliva containing rabies particle. But that saliva will be in a pale brown. So pale brown leads to that it being 100% as a rabies virus particle. Okay, what it contain? That contain rabies viruses. It's a bullet-shaped viruses. It contain 5 different structure protein: G protein, L protein, M protein, P protein, and membrane protein. Different kind of structure protein was a vital role for the attachment, multiplication, propagations here. And okay, here the dog will going to bite in a calf muscles. So immediately that virus to skeletal muscle will travel to the spinal cord. So do the spinal cord reach to the brain. So once reach to the brain is going to affect the entire area is a high and salivary glands, and the entire area was affected. So the virus cannot infiltrate into the skins. Once the virus reaches the brain, it further replicates, resulting presentation of clinical sign from the patient. Okay, you have some doubt. If a person was bitten with dog, if they are not treated properly on a specified time, so it leads to look like as a dog. Why the person was behave like a dog? Because that person entire brain system was fully affected. That brain is a main control of entire our activity for walking or walking and eating, what kind of activities, everything controlled by brain. If the entire virus was multiplied in the brain, there is no proper signal for seeing anything. So going for photophobia, you could not able to hearing, aerophobia, and here also is made up of spasm, that is called a striated muscle. The esophagus is a main mode for the contractions. What materials we are going to take, food materials, everything contraction is going for movement from this to here to there. So there is a signal from brain that entire contraction muscles are not relaxed fully like that. So due to that, you could not able to open his mouth, you could not able to swallow anything, so leads to spasm. So that's why some of the salivation is coming out. There is a drooling of saliva. Next also, here the pharynx and pharynx region. That pharynx and pharynx region, the person you could not able to speak out properly. If you want to speak something, you could not get a very good proper sound and other things, and there is no words. So it leads to this person's making sound. It look like as a dog barking. So that is called as a clinical signs. Then also hydrophobia. Hydrophobia cannot able to swallow anything because of this or non-relaxation of striated muscles. So these are the things was happen if the clinical sign, this person having the spasm, and next is a drooling of saliva, entire area, and also you can find out his hand was tied in a that bed. So this person was in final stage. It been using this oxygen for that nebulization. Next is here what is hydrophobia. You could not able to drink the water, and finally it be in mad condition. So these persons is called as a rabid person because they are in final stage of the situation. Okay, how can we control? So these are the viral production. Viral production previously those person was affected with rabies. In persons too, it was started 1907. From 1907 to until December 2004, we are in the production and supply of sheep brain vaccine or neural tissue vaccine, neural based tissue culture vaccine until December 2004. Excuse me. Using with that sheep, the healthy sheep are inoculated with the rabies viruses, this PV11 rabies viruses. So that sheep brains was collected after the development of symptoms. That that infected brain is going for emulsification using with phosphate, calcium, magnesium free phosphate saline. Then going for inactivated with beta propylactone. That is used as a vaccine. That is unpurified, and it also from that brain some of the unwanted cells. So that kind of things, it leads to the problem to the vaccinated person. That's why we are in the production of tissue culture based vaccine. For the tissue culture based vaccine, so by the using with different kind of surfaces, you are using glass surfaces, or it is in chemical beads as a bioreactor, or it is in roller culture surfaces. Here, some of the pictorial representation is taken from the net. Another photograph will take later. Here. It's a cell culture media. Here is a model of cell line propagations, and during this suitable fixed virus time, a collection of viral harvest. So this is a media. So generally media is two types. One is growth media and is a maintenance media. Growth media, it contains rich amount of growth promoting factors. For example, phytivin, fibronectin, or G protein. These are the protein involvement with cells attachment on the surfaces. You can see the ground is a monolayer form of vero cell line where it's going to attached on the surfaces like so roller bottle. So here roller bottle, we attach the cell lines, immediately the virus is going to inoculate it here. After the inoculation, we are going to the viral harvest. You can see the one thing is a bioreactor. It is main. It's photos taken from my laboratory for the vaccine productions here. This is a replenishing. Here we are using the media, this maintenance media. Maintenance media means the cell cycle. So the normal cell line, cell lines infected with the viruses, cell is being stable. So inside the cytosol, it releases a progeny of different type of G and L protein. If the cells newly synthesized viruses come out, that comes out viruses was present that what do you call it? It is in media. That media replenished with that maintenance media, and maintenance media is going for the collection of viral harvest. That viral harvest here, viral harvest is in every 72 hours. We are collecting the viral harvest. So 4 viral harvests we are collecting. That entire viral harvest we are going to the pooling. So pooling in a single viral harvest. That single viral harvest again going for the concentration by the clarification and also by the IPQC testing process, suitable inactivation using with beta propylactone and tested in animals to check the virulence activity, then going for purification by concentration by the concentration TFF system, tangential flow filtration by ultrafiltration system using with polyvinyl difluid membrane for the nominal molecular weight cutoff is 100 kilodalton. So that 200 liter viral harvest finally you get only 5 liter material. That 5 liter material going for inactivation, then purification by the affinity chromatography system. After the purification, then going for the formulation, then going for filling and filling in a filling vials, then going for freeze dried with the vaccine vial monitoring system. Then we check the quality control test to meet the safety, efficacy, potential of the immunogenicity. That is a quality attributes. So humidity going for formulation. So suitable stabilizer and additives, fill and finish. So random sampling by the quality assurance, sending the national authority the release certificate and supply with a suitable logistic at the end. Why mean by suitable logistic? So that's why we kept in a vaccine vial monitoring. Sometimes, for example, we are going to take the Coca-Cola or any drinks. So while serving, they are using only a +4 degree kept in a fridge. But previous day, what they done? They are open truck, open area, that was travel that. So we could not realize it is not a fridge, but it is not like in a vaccine. That vaccine always once a product produced, that was stored in +4 degree. That entire logistic maintain the same +4 degree at the time of vaccination also should be in +4 degree. Why we need mandatory? Because +4 degree it be in specific for the particular vaccine. Otherwise, it lose that it will be in specific city. It will get down automatic potential will get down. It is not give the or independent protective system, immune system. That's why they things. So that's why this color was happen is a control. Vaccine while monitoring to check the vaccine, it was properly stored, properly transported from the different logistic. So these are the whole viral rabies vaccine production. And also another flow diagram. Same virus line is a master seed, then going for working seed. Parallelly, rabies virus is a master seed. Again going for working seed here. Once a formulation of monolayer, that monolayer cell line going for collected the viral harvest of viral harvest 1, 2, 3, 4 in every 72 hours. That's going for pooled in a single viral harvest. Then concentration by tangential flow filtration. Finally, as inactivation, and inactivation ensured in the animals. And also that going for purification in a affinity chromatography system. That fraction was selected. The selection fraction was removal of the salt by the again dilution filtration. That's a diafiltration. Finally going for clarification using with 0.2245 micrometer filtration. That is called as purified rabies virus protein is the ready to formulation. Either it is a freeze-dried vaccine or is a liquid vaccine. Same kind of process what they done in the world for the production of inactivated COVID-19. So same 3 stages. One is the virus production, inactivation, purification, same virus seeds, virus cell line going for cell factory, clarified and kept in less than 40 degree, around 0.2 to 0.4 micrometer, and also going for cell supernatant and 50 liter, then going for beta propylactone inactivation, that inactivation validation using a suitable animal, then finally that inactivated supernatant going for diafiltration using concentration by buffer exchange, then going for benzonase enzyme digestion to digest the host cellular DNA of virus cell line. After digestion, again going for ion exchange chromatography based on that charge, then going for concentration, then going for removal of the salt by size exclusion chromatography, then 0.22 micrometer filtration, then aluminum hydroxide formulation. That is a vaccine product. So these are the production of whole virus vaccine production of COVID-19. So I'm going to conclude this. Is a little science takes away you from a God, but more of it take you to Him as a Louis Pasteur. Thanking you. Okay. Any doubt?
Participants, if you have any doubt, can unmute yourself or post a question on the chat box.
Yeah, any doubts? Any doubts?
Looks like there's nobody with any kind of doubts. So thank you on behalf of Biotechnology, Oxford College of India. Thank you for accepting our invite. I delivered a rapid lecture introduction. Thank you so much.
Thank you. Thank you. Yes, sir.
Thank you, Jonathan, sir.
Thank you, sir. Thank you, Manjula, sir.
And if you have any opportunities or opening for our faculty to collaborate with your institute, please highlight that so that our faculty can send the students for internship, projects and other kind of activities.
Okay, sir.
Whenever such openings comes, please keep us informed, sir.
Sure, sir. Sure, sir. Whenever coming, I will text you, sir.
Yeah.
And also, we are planning to go for some of the, what do you call, collaboration between industry with institution as a institution. We'll look forward for the future action, sir.
Definitely,
Sure,
sir.
I will
We are
text you,
open
sir.
for all kind of collaboration. And in
Yes.
fact, I will be happy to work with you people. Convey my regards to Dr. Jagannathan also.
Sure, sir.
Okay.
Sure.
Thank you. Thank you very much.
Thank you, sir. Once again, thanks to
Participants, the feedback link is being posted in the chat box and it will be open for the next 10 minutes. And you people can submit your feedback. Sir, you can leave. Sir.
Okay, thank you.
Thank you, sir.
Thank you, sir.
Thank you very much.
Thank you, the team, sir. Thank you, sir.
Leave and the session is closed or now and hello. You can leave, sir, and